Hi everybody, this is Mr. Nolan, and what I would like to do with you right now is to kind of walk through our antibiotic resistance model just to make sure that we're kind of on the same page as far as how do bacteria become resistant. That's our goal, is to be able to develop a model to explain how bacteria become resistant to antibiotics, and we're going to be assessed on this, so we want to make sure that you are able to develop this model correctly. And before we actually look at how we model this, I just kind of want to point out there's a whole wide variety of different ways that a model can look. I mean, if we just kind of cycle through some of the models that we looked at in class, some are better than others, but the important thing is that they all are trying to explain something. They might look different, and like I said, some are better than others. This one is particularly good because we show generations. The student has kind of used color coding to good effect. And so some are better than others, but they all seek to explain how bacteria become resistant to antibiotics. So that's what we want to be able to do today. So to start off, we have to make sure that we know what our model needs to include. The problem here is that we have non-resistant bacteria. We expose them to antibiotics, and what we're finding is that the antibiotics don't work that well anymore. That's sort of the central question behind our unit, is why don't antibiotics work anymore? It's not the antibiotics that are changing, it's the bacteria. So we start off with non-resistant bacteria, and they end up as resistant. So we need both of those things. And that's caused somehow antibiotics are involved with this. So we have to include antibiotics as well. But we've learned over the course of the unit that there's other things involved. There are genes involved here. There's resistance genes. Those genes appear by mutations. So there's other things involved with this model that we need to consider. We also have reproduction and death. These play really important roles in this model. If you don't include these, the bacteria are reproducing and some of them are also dying, then this model doesn't make any sense. So we need to include all these sort of underlined terms in order for our model to make sense. So what we need to start off with is our non-resistant bacteria population. So let's go ahead and throw up that population up here. Okay, so here's our non-resistant bacteria. I'm color coding them red just for, we're just going to choose a color. So we've got our non-resistant bacteria. We also have to recognize that they are being exposed to antibiotics, and then we end up with resistant bacteria. So I'm going to go ahead and add that as well. Okay, so I've included a few basic details here. We know our non-resistant bacteria, through some mysterious process, become resistant. So NR will stand for non-resistant, red, and then resistant will be blue, and that's R. And we know that antibiotics is sort of involved here. So we need to kind of fill in these steps in the middle. There's really more than one. We're sort of going to keep this as simple as we can. I will sort of add an extra step here, but we know this is the skeleton, the outline of our model. We know this is what it needs to look like. Notice that I'm drawing individual cells. You can't just draw a big blob and say that it's our non-resistant population. You can't just draw a big blob over here. We really ought to be seeing the individual cells, because what we have seen is that mutations and genes have to do with this. These resistant bacteria have a different gene than the non-resistant bacteria. Now, there's really only one gene difference, right? They're the same species. They're the same thing, really. It's just like, this is kind of like the human equivalent of, identical twins, only these guys have curly hair, and these ones have straight hair. So it's really a minor difference, the same species. But these guys have this mutated gene, and the non-resistant bacteria have the regular gene that has not been mutated. So where did these resistant bacteria come from? It is tempting to say that antibiotics cause the non-resistant bacteria to become resistant, but that's not quite true. So what's not going on is this. So it's tempting to say that antibiotics are, okay, I've got my non-resistant bacterium, and the antibiotics make that cell into a resistant bacterium. I've got my antibiotics, that bacterium is changing in order to become antibiotic resistant. But this doesn't really fit into our model. This is actually not what's going on. There is something involved here where we have this change, but that's not what's really going on. So you don't want to think that antibiotics turn non-resistant cells into resistant cells. That's not what's really going on. What we have seen is that what's actually happening here is that there are one or two resistant cells hiding out in that non-resistant population. even though this population is non-resistant, there's a few that are sort of hiding out in there already. These particular cells have the mutation. They actually have it already. Right? They have that. The mutation doesn't show up out of nowhere. It's actually hiding in there already. Now, it's only a beneficial mutation to the bacterium as soon as you add antibiotics. The bacterium has that mutation essentially by accident. It's not like the bacterium does anything on purpose. The mutation is already there. It could have been there for millions of years. It's just never really had a use because there's never been antibiotics. So as soon as you add antibiotics, you throw those into the mix, suddenly this mutation is beneficial. That is kind of the key here, that this mutation is only going to be important to the bacteria as soon as you have antibiotics. Okay, so this is a really important idea here, that mutation was already there. Now, mutation, all that means is a change in the gene. So a gene is a stretch of DNA that codes for a trait. That's the idea behind what a gene is. I have genes, you have genes, anything alive has got genes. Even some things that aren't alive have genes. It just codes for a trait. So the presence of antibiotics is what makes that mutation beneficial. The mutation just codes for the trait of antibiotic resistance. Now, there's lots of complicated things we could go into as far as how that resistance works. We don't need to worry about that for the sake of this model. All that we need to do is understand this relationship. The mutation is already there. It's a random change in the gene, but as soon as you throw in antibiotics, suddenly it starts to take on an effect that is absolutely not random. These bacteria are going to survive better than the non-resistant bacteria as long as there are antibiotics. So let's fill in some details here. What is the mechanism by which this model is working? Okay, so I gave us a little more space here. Let's just kind of walk through this. Let's think about what's going to happen here. So our antibiotics, we treat our non-resistant population with antibiotics, and wonderful. So we end up killing a lot of these non-resistant bacteria. We're going to treat them with these antibiotics. So most of these non-resistant bacteria are going to die. So over time, we're going to have reproduction. So some of those bacteria will grow back. Maybe we don't kill every single one. However, we find that those resistant bacteria are also going to reproduce. And suddenly, because you killed a lot of those non-resistant bacteria, you see we have a significantly greater percentage of resistant ones. Before, we only had a couple of resistant bacteria. Now it looks like 50-50. We have even more that are actually resistant. So go ahead and treat them with some antibiotics again. So we're going to use our antibiotics again. And we find once more, well, we're going to kill off most of those non-resistant bacteria, but those resistant ones will remain. So we're going to have reproduction. We're mostly going to have resistant bacteria. Maybe there's maybe a couple of survivors of those non-resistant ones, but as soon as you hit them with antibiotics again, you've killed the last of those non-resistant bacteria. So the idea here is that the presence of the antibiotics sets up an environment in which the non-resistant bacteria are destroyed. They're killed. The resistant ones, in this case the blue ones, the ones that have this mutated gene, are going to reproduce more because they can survive in this environment. So the environment has selected for antibiotic resistance. So what we have here, this is pretty much the most important parts of our model. Our antibiotics set up this environment in which this mutation, which already was there, is beneficial. This random change in a gene, until there were antibiotics, it didn't mean anything to the bacteria. As soon as you add antibiotics, well, a lot of those non-resistant bacteria die, and the resistant ones reproduce faster. So really now in this situation, the resistant gene is better than the non-resistant gene. So we have more of those appearing, and the reason why they appear is because of inheritance. Genes are inherited. That's another sort of important part of this model, is that genes are inherited by the previous generation. So if we kind of jump back and think of our earlier model, we've really got generation 1, generation 2, generation 3, generation 4, in this situation, the gene is actually inherited. Those are things we can add here too. So I know that the screen is maybe a little overwhelming to some of you, but it really boils down to some simple principles of just basic biology. Genes are inherited, and the presence of antibiotic is what makes this mutation, this random mutation, beneficial. And so in a way, the environment, which has got these antibiotics in it, is selecting for antibiotic resistance. It is selecting for that trait. So this trait is better than the non-resistant trait. But this doesn't make any sense unless the mutations are already there. The antibiotics do not cause the mutation. The mutation was already there. This is a very important thing to keep in mind. So it would be an excellent model, but again, there are other ways to illustrate this. Just because I put this up here doesn't mean this is the only way you can show this. You might use colors, or maybe you could use different shapes to show the different bacteria. You don't have to include the generations. You could actually show this in one single step. It really doesn't, we could keep this model very, very simple. However, these are the principles that enable you to show this model. If I only show a big blob and I'm calling it my population, I can't see what the individual cells are doing. So I hope that this is helpful, and that you can kind of now maybe make a little bit more sense out of how do we develop a good model to show how bacteria become resistant to antibiotics.
Drafts rule.